Thanks, Urs, for giving me the opportunity here to talk a little bit about the project, or one of the projects I did during my PhD here. And yeah, it's about aptamers and it's about nanoparticles. I guess you already have some idea about the opportunities and advantages of nanoparticles, but I want to just start the presentation by just talking a little bit about the potential advantages of aptamers, and hopefully I can convince you that there are some. So what's really nice about aptamers, or what are aptamers actually, that's it's single-stranded oligonucleotides, so either RNA or DNA, which fold into a specific three-dimensional structure and then act very similar to an antibody or a peptide, so kind of interact with a specific ligand. What's really nice about aptamers is that they can be selected in an in vitro process called SELEX, and the target range is basically unlimited. So compared to antibodies, that's quite the advantage. So you can select them against small molecules, toxins, or substances that are inherently low immunogenic, but also bigger things like peptides, proteins, bacteria, or whole cells. So the way it works is you start with a semi-combinatorial library of oligonucleotides, incubate it with the target, elute the binding sequences, regenerate it, and then repeat this process a couple of times until you arrive with an enriched library of competent sequences. The work doesn't stop there. You have to sequence them, characterize, chop them down, or modify them. But then hopefully you end up with a defined sequence of an aptamer that falls into such a three-dimensional shape and then has, for example, here a binding pocket for 5-hydroxytryptophan shown here. So yeah, as chemical antibodies, what aptamers are sometimes referred to, they do have some advantages in clinical applicability. They are less immunogenic, or at least so far there are very few toxicity studies about aptamers. They're smaller, roughly 10 times smaller than antibodies, so they have a higher tissue penetration. And again, they can be selected against any kind of target. They also have some advantages in terms of from the manufacturing side. So they are thermally stable. You can heat them up to 100 degrees, and once they cool down, they just fold into the correct structure again. They are much more, or much less susceptible to chemical, or it's easier to chemically modify them using harsher solvents compared to peptides. They're cheap to produce, and they have a shorter selection time. So there's no surprise now that the industry and several companies are really interested in developing aptamer-based therapeutics. There are right now about 50 clinical trials in various stages, both for diagnostic and therapeutic applications. So ranging really from everything from ocular diseases, infectious diseases, and of course to oncology. And the application really is similar to antibodies. You can use them for therapeutic applications, targeting, drug delivery. And what we are really interested in is bioimaging. So this is the last bit of introduction. There's a specific aptamer I want to talk about called SGC8. This aptamer was originally selected against a lymphoblastic leukemia cell line. Has a very high affinity against protein tyrosine kinase 7. So that's the molecular target of this specific aptamer. And this PTK7 is overexpressed in a range of cancers. It was first developed or discovered in colon carcinoma, but it's probably in quite a few cancers it's overexpressed. So this SGC8 aptamer is among the diagnostic ones. It's really one of the better described or more used ones. So you see here in the slide, takes up a large proportion of all the molecular targets is PTK7 and this specific aptamer. And actually, not too long ago, there was the first in-human trial of a radio-labeled aptamer, of a gallium-68 labeled SGC8, that was injected into a colon carcinoma patient. And you can see here that the pharmacokinetic profile of this aptamer is actually really benign or really favorable for imaging, very similar to peptides, for example, which are already used in clinics. So there's very low uptake into off-target tissue. And really only any activity is in the excretory organs, so in the kidneys. However, unfortunately, in this study at least, there was a very low tumor uptake. So this now finally brings me to our idea for this project that I'm going to talk about. So we wanted to combine this kind of advantage of aptamers being highly specific to a molecular target with the advantages of nanoparticles, so the ability to circulate for a long time and then hopefully have a better tumor retention and a slower washout. So we used, we wanted to use HBG, or hyperbranch polyglycerol, as our carrier in this study. Used an aminated version of HBG and reacted it with simple amide coupling with a transcyclooctene, or TCO. And then we could use a very simple click chemistry approach to just click any kind of functionality that we wanted to onto this HBG TCO. So simple here really means it doesn't require any special kind of solvents or temperatures or reagents. So we use, for example, a fluorophore-labeled tetrazine, a radio-labeled tetrazine, or an Aptamer functionalized with a tetrazine, and just click it onto HBG. So in terms of characterization, this click chemistry approach based on the tetrazine ligation afforded us a very convenient and quick way to produce this radio-labeled Aptamer HBG probe here with very high purity. We also did some studies on size and surface charge. You can see here in red, with increasing functionality or increasing functionalization of HBG, we see a decrease in surface charge, which corresponds to the negatively charged backbone of the oligonucleotides here, and an increase in size with increasing functionalization. We also did some gel electrophoresis to kind of show that we are actually able to functionalize HBG, this is shown here on top, with our oligonucleotides, so SGC8 aptamer, or a control sequence, which overlaps very nicely with our activity or our fluorophores. So this synthesis approach worked really nicely. And one big problem of aptamers is they are small, but they are also very susceptible to nuclease degradation. So we were really excited to see that by just conjugating SGC8 to HBG, we were able to really protect it quite significantly from protein, or nuclease degradation. So that was very nice to see. We also did some very preliminary toxicity studies. We did not expect anything. Both aptamers individually and HBG never showed any signs of toxicity, and we did not observe anything in our hands as well. So next we were interested if those aptamers actually retain binding affinity after conjugation to HBG. So here in this top graph here, you see HBG SGC8, HBG scrambled, which is just a randomized control sequence of this aptamer, incubated at different concentrations with hex cells in this case. So there was no binding at all. But on the other hand, with HBG SGC8, we saw a nice concentration-dependent shift in fluorescence. We also checked whether HBG itself would stick to cells or bind to the cells. So we did a titration there against cells. And we only observed a little bit of binding at very high concentrations that are way beyond the concentration that we are actually using in vitro or in vivo. I just want to mention very briefly, at higher concentrations, so at 500 nanomolar relative to the aptamer concentration, we did see a little bit of non-specific binding of HBG scrambled, which is not ideal, and that's, I think, a general problem of aptamers, and I'm happy to talk about this in more detail later. But let's leave it at that for now. And yeah, you remember we have a HBG transcyclooctene functionalized nanoparticle. So we can actually use this for pre-targeted labeling, and ideally at a maybe much later stage for in vivo pre-targeted imaging. So the way it works is we incubate cells with HBG SGC8 or HBG scrambled, which has those free TCO functionalities. Let it incubate, and then add in a second step a tetrazine functionalized with a fluorophore, Cy3 in this case. And we really see some nice extracellular membrane staining here of HBG SGC8, but not for the scrambled version, confirming that we have specific binding to this extracellular target here. We also did a saturation binding assay of our radio-labeled HBG SGC8. In this case, this was against the breast cancer cell line. And our affinity in our hands was around 4 nanomolar, which is on par or in the same order of magnitude as free SGC8. So overall, we were very happy to show that HBG SGC8 does retain binding affinity, and so in vitro those results looked really convincing. So now moving on to the in vivo evaluation. So we looked into the tumor uptake and overall pharmacokinetics of HBG SGC8, HBG with the scrambled aptamer sequence, and then just naked HBG without any single-stranded oligonucleotides there. So after intravenous injection into A431 xenografts, we saw what we basically expected for a nanoparticle formulation, high activity in the blood pool, it's even visible in the carotid artery here. And then with increasing time, we see increasing uptake into the liver, or basically the terminal fate of most nanoparticles is the liver. And we're also very happy to see quite decent tumor uptake here. Overall, the pharmacokinetics is as expected for HBG-based formulation. We have a very long circulation half-life, so instead of a few minutes for a free aptamer, we are now in the realm of hours or days even, and we have relatively high liver uptake. And overall, no difference between the different formulations. If we look into the tumor a little bit more detailed, and this I guess comes into the direction what Colin said already, how in vitro translates to in vivo results, we definitely saw a little bit of a trend in terms of tumor uptake. So our HBG scrambled showed a lower total uptake, but it was not statistically significant compared to the other formulations. And especially here, the blue and green line, HBG SGC8 and the naked HBG is very similar. So similar in the organ distribution after 48 hours. We do see a little bit of a trend, a lower activity in the tumor for HBG scrambled, but overall no statistically significant difference. So unfortunately, we had to conclude here that at least with this tumor model, there was no significant difference between the formulations. HBG SGC8 was not able to accumulate higher in the tumor or retain or stay there for longer. So I guess we have to conclude that in this case, at least the EPR effect or enhanced permeability and retention effect of nanoparticles was kind of the dominating or the driving factor here in the overall tumor uptake. And with this, this brings me to the conclusion. So we developed a very nice click chemistry-based synthesis scheme, which is kind of could be used as a platform. It's very easy to just replace, for example, this SGC8 aptamer with any different kind of aptamer. To create this nanoprobe. We have shown that HBG conjugated aptamers are protected from nuclease degradation and have shown excellent binding in vitro, but unfortunately could not show the same superior effect in vivo. So I guess the next step then would be to perhaps try this in a different tumor model or try a different aptamer. And with that, I want to thank Urs and all the lab members that helped with this project, and then also my co-supervisor in Copenhagen, and then especially also Charles Haynes, who is kind of an expert in this development of or selection of aptamers, who was also very helpful for this study. And finally, funding from the Lundbeck Foundation in Denmark. And with that, I want to thank you, and I'm happy to take questions. Oh, sorry. Yeah.
Peter.
I would say you're doing pretty well. You've got the targeting ligand and you have the same circulation lifetime and the same amount as getting to the tumor site. That's when you don't have targeting ligand. That's pretty unusual. Usually when you put target information on things, you have to control that more quickly. But presumably you're hitting some different cells in the tumor. Have you looked at that?
Yeah, we have done some histopathology. So I think I haven't shown this here, but one of the problems with this specific A431 tumor was that the entire core is very necrotic, or, well, not perfused. And yeah, of course, I mean, the results for imaging studies depend on the perfusion of the tumor. So that's why I'm saying maybe it works with a different tumor model, but yeah, we have to see.
As lot of other people have hit if you look in the licensing literature.
Yes.
What was the molecular weight of your polymer?
Sorry. Yeah, this was a 700 kilodalton HPG, so roughly around 10 nanometers in size.
Yeah,
And then, I'm not entirely sure. I have to look this up.
Very low.
Yeah.
1.0.
One.
1.01.
Thank you,
Yeah,
Kathy.
so the hydrodynamic rate has changed from your core polymer is about 5 up to maybe 10, 12-ish.
Yes. Yeah.
So that's not necessarily size per se.
Yes.
That's not really what it's measuring. But what would you estimate to be the actual particle size based on other techniques?
Yeah. So on other techniques, that's kind of, I guess the closest one would be gel permeation chromatography. There are some standards. That's why I'm saying around 10 nanometers for about 500 kilodalton HPG is a number that is around in the literature. So, and I agree with you that it's not size, but it shows a trend, right? And similar with the charge. So.
Okay, Peter has one more question.
One thing to think about would be the target things that are in the circulation using your approach. I think you have to look at that. Targeting T cells, targeting.
Yes. Yeah, I mean, with HPG, I mean, we will always have high liver uptake. So that's why I said it would be interesting for pre-targeted imaging, potentially in the future. But yeah, I agree. I'm not sure about direct imaging of nanoparticles for tumors. No, it's going to have a very hard time in the clinics.
One more.
I was wondering if you could expand this technology beyond STC.
Yeah, for sure. I mean, so the way we created those aptamers, you can very easily order aptamers functionalized with a free amine group. We attach this to a tetrazine and click it onto HPG. So we could, in theory, synthesize any kind of aptamer, or any kind of peptide, small molecule, really.